Hi everybody, this is your TF Mary, and this is a supplemental video on protein structure and how it changes during cooking. So some of these words in the title might not be so familiar right now. Denaturation is just another word for unfolding, how a protein loses its folded structure. And coagulation, I don't know, maybe this makes you think of, clumping in milk or the ricotta cheese that you're going to make during lab in the next week or two. You'll learn all of these terms a little bit better as we go through this video. So what you're looking at here is a close-up view of a protein that has been unfolded and stretched out as far as it would go. You learned in class that a protein is a polymer made up of amino acids, and each amino acid here starts with a little blue nitrogen and ends with a carbon double bound to a red oxygen atom. So each of these amino acids has a backbone that is exactly the same as the others, but each amino acid also has its own side chain that has a different shape and different charge and polarity. So let's take a closer look at a few of those. So let's start with this amino acid over here on the left. It's called lysine. And one thing you're going to notice as you compare that to the structure here on the right is that none of the hydrogen atoms are shown. When hydrogen atoms are shown in structures, they're normally white in color. In proteins, we normally don't show them because they take up so much space and make it so difficult to see what else is going on. This amino acid has a positive charge, which you can see indicated down here. That would allow it to participate in ionic bonds, which are a very strong type of non-covalent bond. We'll talk about it a little bit later. So next let's take a look at this amino acid called phenylalanine. If you take a look at the side chain, you can see that it only has carbons and hydrogens in it, and that makes it very nonpolar. So this is an amino acid side chain that would like to be away from the water, which is what the protein is floating in. That means that when the protein folds up, this amino acid will probably wind up on the inside. Okay, so now I've zoomed out so you have a bird's eye view of the whole protein. For reference, earlier we were looking just at this lysine and phenylalanine here. The typical protein has around 300 amino acids in it, and as you can see they're all linked together through one long chain. There is one additional covalent bond right here. And now if I zoom in on that spot, you can see that that extra bond is formed between the sulfur atoms on two cysteine amino acid side chains. So this is an interesting type of covalent bond in that if you lower the pH, you can actually break this bond, whereas normally a covalent bond is very stable. Okay, so now we're taking a look at the protein in its folded state. Instead of being stretched out end to end like you saw earlier, now that long amino acid chain is curled up on itself. And if you're like me, you look at this and you can't figure out where one end is or the other, and it's hard to tell what's actually going on here. So what I'm going to do is show you where that backbone is, that long thread that connects all of the amino acids together. In this representation, you can start to see that the protein is doing a lot of interesting things. That long chain of amino acids is sort of curling in a helix, or it's going back and forth in strands. But it's still very difficult to see what's going on because of all of those side chains, so let's hide those as well. Okay, so now we can finally start to see where this protein begins and where it ends. If you gave me a piece of string and asked me to fold it up like this protein, I would have a really hard time. But somehow, inside of a cell, the protein folds up correctly every time. And that's because it's stabilized by these extra non-covalent bonds that help it retain its shape once it folds correctly. One of the major contributors to the protein's stability is hydrogen bonding. And when you see in a cartoon representation either arrows that look like this, or helices that look like this, what that's trying to tell you is that there are a lot of hydrogen bonds in a very particular orientation there. And in order to see what those look like, we're going to have to switch back to the stick model. Okay, so that got visually complicated pretty fast, right? It's difficult again to see what's going on. You learned earlier that these amino acid side chains do a lot of interesting chemistry, but here the hydrogen bonding is actually between the atoms in the protein backbone. So I'm going to hide the side chains again. Okay, so now we can finally start to see something that looks like that curlicue cartoon that you saw earlier. And there are no hydrogen bonds, remember, shown here, but each of these blue nitrogen atoms has a hydrogen bond attached to it. And that's forming a hydrogen bond with an oxygen above. I'll draw in the bonds now. There were actually a lot of hydrogen bonds there. And remember, this is only one small portion of the protein. In order for the protein to unfold, each of these hydrogen bonds would have to break, which is energetically costly, but then since the protein is floating around in water, new hydrogen bonds will just form with the water. So this doesn't create a really big energy difference to help the protein stay folded. We need to look at some other bond types, like ionic bonds. So here's our eagle-eye view of the protein again, and this time I'm going to zoom in on this part of the picture where there is an ionic bond. I had to hide a few amino acid side chains to make this more visible, but the bond that we care about is between this oxygen, which is on an amino acid called glutamate, and a nitrogen on this amino acid called arginine. Now that probably doesn't look very different from the hydrogen bonds you saw earlier, but if we look back at the structure we can see that actually each of these atoms is charged, and they have opposite charges. So this must be an ionic bond. This is a very strong type of non-covalent bond. There's another major force that helps the protein stay folded called the hydrophobic effect. This protein would be floating around in water, typically inside of food, and the amino acids that have side chains pointing towards the outside of the protein are mostly hydrophilic. That means they like to interact with water. But the side chains that point towards the center are mostly hydrophobic. They can't form hydrogen bonds with water, so it's energetically favorable for them to be stuck in the center of the protein near each other. So I guess the take-home message for this part of the talk is that once the protein folds up, there are hundreds and hundreds of interactions like hydrogen bonds, ionic bonds, nonpolar interactions that keep the protein folded that way. So now let's talk about how we cause proteins to unfold during cooking. One of the ways that we cook foods is by raising the temperature. So I've drawn here two amino acids. They're involved in an ionic bond, and on paper obviously they're not moving, but in reality there would be some movement of all of the atoms in each of these amino acids. And as the temperature goes up, the motion goes up too, and that will eventually tend to break this ionic bond. Once the bond is broken, the two amino acids can start to float apart from each other, and that's how the protein begins to unfold at high temperature. So another way we can break the ionic bonds, besides raising the temperature, is by lowering the pH. It turns out there's a reaction between the protons that we add when we lower the pH and the negatively charged side chains on amino acids like glutamate and aspartate that participate in ionic bonds. And that reaction results in a neutralization of the charge. Once it's neutralized, that amino acid can't participate in an ionic bond anymore. And the more of these protons we add, the more we push the reaction over to the right and get more things that look like this. Since we're reviewing equilibrium constants this week, let's talk a little bit more formally about what it means for the reaction to go to the right when we add more acid. So normally when the reaction is at equilibrium, that means it's been happening for a long time and there are as many reactants being converted into products as there are products converting back into reactants. There's a certain ratio between the concentrations for those different things, and that's determined by the equilibrium constant. So this is it turns out to just be 10 to the 4.5 for this particular reaction. So what happens right after we add more acid? The concentration of protons has gone up, and there's no time for the reaction to reach equilibrium again, so that means that this whole ratio will be less than the equilibrium constant. And in order for this reaction to get back to equilibrium, some of this stuff in the denominator is going to have to convert back to stuff in the numerator. And that's how we can tell what the reaction is going to go from the left over to the right. Another technique we commonly use in cooking to disrupt protein folding is just adding more salt to the food. So here again you have two amino acids involved in an ionic bond, and we're going to add table salt, sodium chloride, that's NaCl. And when the salt is added, it dissolves to make two oppositely charged ions, and those ions can substitute for the pairing that you had here so that you wind up with more ionic bonds, but they're not between amino acids anymore. And that will allow the amino acids to float apart from each other and lead to the protein unfolding. You already saw one way that pH disrupts protein folding, by neutralizing charge, but there's another way that's very similar to salt. Here we've just added more protons, and the protons are pairing with the negatively charged amino acid to disrupt an ionic bond. So why does protein unfolding cause foods like eggs to become solid when we cook them? Well, if you imagine proteins that are floating around in their folded state inside of a cell, most of the side chains on the amino acids are forming intramolecular bonds. That means bonds within the proteins and not between proteins. That lets the proteins move relative to each other. When you unfold the proteins, there's a good chance that they'll instead start bonding to each other instead of reforming the bonds that they originally had within the molecule. And that tends to create a sort of tangled web, almost like a matrix or a gel of proteins within the food. So we'll talk about this again a little bit later in the week on elasticity. But for now, keep in mind that when you unfold proteins, they can come together to form a mesh that makes a food more solid.